Hello students, welcome to EPG Pathasala. I am Dr. P. Nagaraj, Assistant Professor from Anna University, Chennai. Today we are going to talk on module Protein DNA Interaction 2 from the paper Structure and Function of Biomolecules. So today objective of today's lecture is Introduction of Protein and DNAs, Basic Function of Proteins and Basic Function of DNA, and specific and non-specific interaction between protein and DNA. Of course, the importance of those interactions we are going to study in details. Why to study proteins? Although DNA are the message of the cell, proteins are the medium through which message is expressed. One means little without the other. What function the DNA protein interactions are involved in? DNA replications and DNA repair, whereas DNA recombinations and transcription, etc., these are all the functions that DNA can involve. Identification of interactions, we concentrate on the molecular basis of protein DNA complexes known at high resolutions. Two effective techniques are already available. One is X-ray crystallography and the other one is NMR spectroscopy. Both are equally valid, but neither is sufficient without the detail of kinetics, thermodynamics and site-directed mutagenesis studies. So using those all together, we can able to understand what kind of interaction which is present in proteins and nucleic acids. History of structure determinations. Structure of DNA is regular. A list of the positions of the atoms in the double helix, the stability of DNA and the Chargaff rules, and provided a model for how DNA stores genetic informations. Proteins are much less regular. It is more difficult to understand. The first structure of DNA binding proteins were of the stable and abundant nucleus binding to single strand nucleotides. Interaction in early, again protein DNA interaction. Work on more complicated protein DNA complexes, overexpressing normally scarce proteins and of synthesizing large amount of oligonucleotides. Only in the late 1970s did it become reasonable to try to determine the structure on oligonucleotides, the protein it interacts with and the complex between two. There are few people got Nobel Prize working on protein DNA interactions. Aaron Clark in 1982 and Roger Kornberg, Alex Rich on 2006. The forces between protein and nucleic acids. There are four major forces that can occur when proteins and DNA interact. Changes in free energy of association to specific interaction between protein and DNA. Four major forces between protein and nucleic acids are electrostatic forces, also known as salt bridge, dipolar forces, also known as hydrogen bonds, entropic forces, the hydrophobic effect, and the fourth one is dispersion forces, base stacking. These are all the four kind of major forces between protein and nucleic acids. Electrostatic force, salt bridge. Electrostatic force are long range, not very structure specific, and contribute substantially to the overall free energy of associations. Salt bridge are electrostatic interactions between the groups of opposite charge that typically provide 40 kJ per mole of stabilization per salt bridge. In protein DNA complexes, they occurs between the ionized phosphates of the DNA and either delta ammonium group of lysines, the guanidium group of arginines, or the protonated imidazoles of histidines. These are all the possible way the protein DNA interactions or complex can occur. Electrostatic force, salt bridge again, changes in sequence of bDNA perturb the average structure only subtly. Salt bridges alone therefore cannot distinguish one bDNA sequence from another. Patterns of salt bridge, however, could clearly be used to distinguish single strand from double strand DNA and B from zDNA. Dipolar forces, also known as hydrogen bond. Hydrogen bonds are dipolar, short-range interactions that contribute little to stability of the complex but much to its specificity. Hydrogen bonds occurs between the amino acid side chains, the backbone amides and carbonyls of the proteins, and the bases and backbone sugar phosphate oxygen of DNA. That is where the hydrogen bond possible. Dipolar forces, again, when protein DNA molecules are not complexed, all their exposed hydrogen bonds donors and acceptors form linear hydrogen bonds to water. Hydrogen bonds are results of dipole-dipole interactions will make the hydrogen bonds. So the substrate, which is both having hydrophilic and hydrophobic part, whereas the enzyme, the outer layer is hydrophilic in nature. So water can interact with substrate and enzyme in case bringing together, connecting together to make the enzymatic reactions or interactions. Dipolar forces, again, when the complex forms, there is a little change in the free energy due to hydrogen bond formation. If the linear hydrogen bonds to water are replaced by similar one between the macromolecules. By contrast, forming bent hydrogen bonds carries a free energy penalty up to 4 kJ per mole hydrogen bonds. The hydrogen bonds are very important in making sequence-specific protein DNA interactions. Entropic forces, the hydrophobic effect. Hydrophobic nothing but is a nonpolar effect. The hydrophobic forces are short range, sensitive to structure, proportional to the size of the macromolecular interface, and contribute to the free energy of association. These are all the factors. Apart from that, the hydrophobic effects is due to the behavior of water at an interface. When molecule aggregates, the order water molecules at the interface are released and become part of the disordered bulk water, thus stabilizing the aggregate by releasing the energy of the system. Molecules of water left at the interface between the protein and DNA obviously decrease the entropy of the system. Consequently, the surface of the protein and DNA tend to be exactly complementary so that no unnecessary water molecules remain when the complex forms. However, specific water molecules with defined functions in sequence recognitions are often bound in the interface. Dispersion forces, base taking. Dispersion forces have a short range but are very important in base taking in double-stranded DNA and the interactions of protein with single-strand DNA. That is very important. That range is very short. Base taking is caused by two kinds of interactions, the hydrophobic effect and the dispersion force. These two forces will be involved in case of base taking effect. Dispersion forces via base taking. Molecules with no net dipole moment can attract each other by a transient dipole-induced dipole effect. It is very sensitive to thermal motions of the molecules. For double-strand DNA, dispersion forces are clearly important in maintaining the structure by base taking. For single-strand DNA, they also help it to bind protein because aromatic side chains can intercalate between the base of single-strand DNA. The strength is proportional to dipole-dipole distance. Geometric constraints imposed by the nucleic acids. All DNA have repeating polyionic backbones, and so all proteins that binds them have strategically placed arginines and lysines that create an electrostatic field to neutralize the negative charge. Contracts to the base are called direct readout because what contract from depends directly on the sequence of DNA. Distinguishing sequence by how the sequence affects the distortability or conformation of DNA is called indirect readout. There are two types of readout that is based on the interaction between nucleic acids and proteins. Double-stranded bDNA. Simple model building predicted two of them many ways in proteins interact with bDNA by hydrogen bonding. There are two types. Number one, an antiparallel beta sheet interacting to the phosphate backbone in the mirror groove. Second one is an alpha helix interacting with the base in their major groove. The pattern of sites are more dissimilar for each base pair in the major than the minor groove. So this is what happened in double-stranded bDNA. To distinguish cognate sequence from all others by direct readout alone, protein must form more than one hydrogen bond to some of the base pairs in the major group. So protein always making hydrogen bond with either nucleic acids, it may be more than one. In specific proteins, bDNA complex about half of the hydrogen bonds are to the bases and the other half of to the phosphate backbone. So the protein making hydrogen bond, one is with nitrogen base where more electronegative nitrogen atoms available, other one is in the possible with this phosphate backbone. Why? Because phosphate also having oxygen which is more electronegative which can make a hydrogen bond. Single stranded DNA, hydrophobic bases in single strand DNA are more exposed. Single strand DNA BP has more hydrophobic DNA binding surface than DNA AB. That means this DNA BP is more hydrophobic, that means it won't attract water or it will be more like to nonpolar phase. The hydrophobic surface often contains aromatic group which is interact more effectively with DNA bases and also an electrostatic field that neutralizes the charges of the phosphate backbone. This is very important. The hydrophobic part which is basically made up of aromatic group which have more close interaction with nonpolar part, whereas the polar part it won't have any kind of interactions. Also the charges can be neutralized by electrostatic field. Possible because the structure of RNA varies more than of DNA. Protein seems to recognize RNA is more way than the recognized DNA. DNA RNA having the difference between the ribose sugar where the second position of carbon having the hydroxy group will give me RNA. If doesn't have hydroxy group, that is called DNA. RNAs even more than DNAs may be recognized by indirect readout. The correct RNA can distort to fit the protein whereas the incorrect one cannot. So this is what the correct RNA easily can go and fit in the proteins. If doesn't have, that won't be fit into the proteins. The kinetics of forming protein nucleic acid complex. Two factors affect the rate of formation of all protein DNA complex: random thermal diffusions and long range directional electrostatic attraction. These two are very, very important phenomena. A one-dimensional random walk can account for the observed rate of genome sequence specific protein DNA complex. The protein first binds non-specifically to the DNA and then diffuse or jumps along the DNA until it finds the appropriate sequence. So this is how the kinetics can be measured via protein nucleic acid interactions. All sequence specific DNA binding proteins may bind DNA in two ways, one for tight sequence specific binding and the other for looser non-sequence specific binding. This is how two way it can bind. Protein nucleic acid interaction again, which is a non-specific interactions. Another one is a specific interaction. First we look at details of non-specific interactions. Non-specific interactions are single-strand nucleic acid binding proteins and non-sequence specific nucleases, polynucleotide polymerases. These are all comes under non-specific interaction. That means it won't be site-specific. It will interact randomly to the protein and nucleic acids. The need for packaging. The fundamental building block of chromatin in eukaryotes is the nucleosome, a protein DNA complex. The nucleosome core particle consists of 146 BP of DNA and 8 small highly basic histone proteins. The DNA wraps around the histones out octamer to form a negative supercoil. So bacteria also use small basic proteins to package DNA such as dimeric HU proteins from E. coli, whose long beta strand arms presumably wrap around a double-stranded DNA molecules. Viruses are highly symmetric particles that can pack their nucleic acid genome efficiently inside the protein capsid. Protein subunit containing many basic amino acids interact with the viral nucleic acids in a non-sequence specific manner. In the helical TMB, some sequence specific contracts are involved in directing assembly of the virus, but there are no such contacts in the icosahedral FHV. Single-stranded nucleic acids binding proteins. Single-strand nucleic acid is formed during replications and most organisms produce protein to bind it. These proteins form an important but diverse group, but with the exceptions of gene 5 protein from bacteriophage. There is a little structural information on how they interact with DNA. A model has been suggested in which lysines and arginines neutralize the DNA phosphate backbone and the bases stake against aromatic amino acid side chains. This is the beauty of single-strand nucleic acids binding proteins. Non-sequence specific nucleus. All organisms must degrade DNA during their life cycles. There is no one enzyme designed for this purpose, but rather a large number of enzymes with different specificities. This includes exo and endonuclease and enzyme specific for single-strand and double-strand DNA and for base sequences. Example, RNase and DNase. That means this non-sequence specific nuclease which can bind either single-strand or double-strand DNA without any specific. RNase and DNase have different reaction mechanism because RNase use the ribose 2-days hydroxy group which is not present in DNA to attack the 5-days phosphate ester linkage. So this is how that RNase and DNase varies, because in RNase the 2-days hydroxy group present, whereas in DNase there is no such a 2nd position having the hydroxy group in the pentose sugar. Ribonuclease A, varnase and binase. RNase A is not sequence specific because it is only interact with the base at the active site. All other contacts are electrostatic ones to sugar phosphate backbone. This is how the ribonuclease have interact with proteins. Deoxyribonuclease 1. The DNase 1 cleaves different sequence with the different rate because of sequence dependent steric hindrance at the active site. Guanine to cytosines tracts accommodate the catalytic loop better because they have wider minor grooves than the adenine and thymine. So always guanine cytosine having 3 hydrogen bond between each other, whereas in adenine thymine always have 2 hydrogen bond with each other. This will make more comfort. Polynucleotide polymerases. There are 4 classes of template directed polynucleotide polymerases: DNA or RNA dependent and DNA or RNA polymerizing. All add nucleotides to the 3 days end of growing polynucleotide chain, but they differ widely in how accurately they replicate the DNA and how many nucleotides they add before associating. That means the DNA which is binding together, and that will replicate how many numbers which is present in the molecule. DNA dependent DNA polymerases. Example, E. coli DNA polymerase 1 and 3. All cellular DNA dependent DNA polymerases have 3 days, 5 days proofreading exonuclease, require a primer to begin synthesis and replicate their own DNA the most faithfully. The Clino fragment of pole 1 contains 2 widely separated domains, one carrying the polymerase activity and the other 3 days, 5 days proofreading exonuclease activities. That is very, very important. These 2 can be monitored by DNA polymerases. The DNA approaches the polymerase from nucleus side and bends by 90 degrees to enter the polymerase site. The protein doesn't read the DNA sequence at all. Instead, when an incorrect base is added, the DNA strand separates and the daughter strand is therefore more likely to reach over to the exonuclease, which then removes the incorrect base. This is how the DNA interact with the proteins. RNA dependent DNA polymerases. HIV-1 reverse transcriptase or RTase. The RTase, which is nothing but a reverse transcriptase, is a unique heterodimer because its 2 subunits have same sequence yet fold differently. The P66 subunit fold into polymerase domains and transcriptase H domains. The DNA in the complex is A form near the polymerase active sites. Near the active sites, the palm and thumb hold the primer strand, while the palm and finger hold the template strand. Two polypeptide helix interact with the phosphate backbone and probably ensure the transcriptase tracks the DNA correctly. Protein nucleic acid interaction, which is specific interactions, which is described in slide number 43. Specific interactions are the placement of alpha helix in the major groove appears to the most common way of recognizing specific DNA sequences. Other part of proteins which form hydrogen bonds and salt bridges to the DNA backbone position the element on DNA so that it achieve recognitions. So this is how we can estimate the specific interaction. There are many types which is available. Direct readout of DNA sequence, most often in the major groove, is an important part of sequence specific binding, but it is by no means the only component. The direct readout can involve hydrogen bonds directly to side chains, to the polypeptide backbone, or through water molecules, or depend upon hydrophobic interaction. So these three things very important in case of direct readout. Number one is directly to side chain, it may react with the side chain, or to the polypeptide backbone, or the water molecules, depend upon hydrophobic interactions. Indirect readout is also important. The correct DNA sequence may differ from canonical bDNA in a way that increases the surface area buried, the electrostatic attractions, or the number of hydrogen bonds formed. That can be explained via indirect readout. The protein can also changes conformation upon binding, affecting the overall stability of the complex or the ability of the protein to recognize a specific sequence. Oligomerization upon binding the correct sequence, as in MET repressor, GCN4 and glucocorticoid receptors, often increases affinity and specificity. Consequently, there is no universal code by which protein recognizes DNA sequences. Even a single family such as HTH proteins, the recognition helix is presented to the DNA in many ways. So there is no method or there is no universal code which is available for protein recognizes DNA sequences. The need for specificity for a cell to function at all, protein must distinguish one DNA from another very accurately. Then only the cell function will happen. Proteins that bind specific DNA sequence also bind non-specific ones. In some cases, like transcriptional regulators, this binding is intrinsic to function. In others, like the tRNA, the binding is merely unproductive. So the binding is very important to carry out all the biochemical reaction. It's a site specific and binding is very important. If it is wrongly binding, then there is no reaction or no other interactions. Quantitative specificity. The simplest model of a protein DNA binding reactions is given by the equations T plus xi gives T dot xi. Here T is free proteins, xi is any one particular site or non-site DNA, and T dot xi is bound complex. That means a free protein react with DNA to give protein DNA complex. A convenient way to quantify the specificity of such a protein is to normalize the binding constant. So binding constant nothing but K equilibrium, the reaction rate, will give T dot xi divided by T into xi. That means this is how to some user defined reference values can be mentioned here. Binding constant can be normalized to an average of the set of K equilibrium over all the xi yielding specific binding constant Ks. Assuming that the position in the binding site contribute independently to the binding energy, a table of Ks values equivalently delta Gs equal to minus RT ln Ks for all the single mutants of binding site allows one to estimate the affinity for any site or non-site sequence. This is how the final rate of reaction can be mentioned. Free energy, change in free energy is delta G is equal to minus RT ln Ks nothing but specific binding constant. One standard method of measuring binding affinities uses a gel shift experiment. So which is again K into Xi equal to T dot Xi T into Xi. Typically the ratio of bound to unbound DNA is determined at several different proteins concentration, and then curve fitting is employed to give a best estimate of K into Xi. Multiple determinations of same constant indicate that this approach can give values that are accurate to within the factor of about 2. This is how we can mention. Transcriptional regulators, the helix turn, helix motif. The prokaryotic complexes and eukaryotic complexes, the homeodomains. These are all the things. Other alpha helical binding motifs, beta sheet binding motifs. The MET repressor family, the TFIID, TATA box binding proteins are important class of these proteins. Restriction endonuclease, echo RI and echo RV, that means echo R1, echo R5. Echo R1 and echo R5 have very different structure and interact with DNA differently. The former, that means echo R1, only in the major groove, the latter, that means echo R5, both grooves. However, both employ the same enzyme mechanisms and catalytic residues, and both achieve their high degree of sequence specificity similarly. They complex with the cognate DNA in the highly cooperative and symmetric manner. In the complex with the cognate DNA, much the free energy of binding has been used to drive the cognate DNA into an unfavorable conformations that places the cis phosphodiester bond, the active site, and the complex, the binding site for the essential magnesium 2 plus. Enzymes that flip out nucleosides. Nucleosides flipping involves rotations of backbone, bonds to expose to out of stack base, double strand DNA. It can then be substrate for enzyme catalyst chemical reactions. Rotation about alpha, beta, gamma, delta, torsional angle but not the glycosidic bond appears to be required. The phenomenon is fully established for 2 restriction methyltransferases. There is a strong evidence for nucleoside flipping for 2 keys repair enzyme. Other examples are emerging, and the phenomenon is likely to prove general for enzymes that requires access to unpaired bases. It appears to need no external source of binding energy. So students, let us know, summarize what we learned from this module. We learned introductions of proteins and DNA, and basic functions of proteins and basic function of DNA, and non-specific interaction also we have studied, and specific interactions also we studied. Apart from that, what is the implication we achieved from those interactions also we studied in details.